Hey everyone, we're Group 15, which is Bryan, Michael, and myself, Athena. Our project is on the paper Remodeling of the Binding Site of Nucleoside Diphosphate Kinase Revealed by X-ray Structure and HD Exchange. The paper discusses research on the nucleoside diphosphate kinase, or NDPK for short, from the pathogen Mycobacterium tuberculosis, or MT for short. MT is the causative agent of tuberculosis. To start off, we're going to go over some background information on kinases. Kinases are extremely important and are involved in a number of different functions, including metabolism, protein regulation, and secretory processes. A kinase is an enzyme that catalyzes the transfer of phosphate groups from high-energy phosphate donating groups, like ATP, to substrates. The process is done by phosphorylation, which is drawn here. Our paper focuses on a special type of kinase, nucleoside diphosphate kinase, or NDPK for short.
Nucleoside diphosphate kinases are enzymes that catalyze the transfer of the terminal gamma phosphate between nucleoside triphosphates and nucleoside diphosphates, maintaining an equilibrium between the phosphorylated nucleosides. The major phosphate donor is ATP and the acceptor is GDP. The transfer is a non-sequential, reversible, double displacement reaction in which the NDPK is momentarily phosphorylated on a histidine residue, forming a phosphoenzyme intermediate. In addition to its main function, NDPKs have a number of secondary functions, including protein histidine kinases, performing a 3 to 5' exonuclease activity, and playing a role in embryonic development.
The NDPK that was discussed in our paper was Mycobacterium tuberculosis, NDPK, or MT-NDPK for short. Tuberculosis is a potentially serious infectious disease that mostly affects your lungs. In tuberculosis patients, thermal-stable proteins, including NDPK, prevent the degradation of MT by macrophages by blocking the fusion of the lysosome with the phagosome.
This is one of the reasons that TB is so difficult to fight on a cellular basis. So why can MT-NDPK evade degradation? The exact process is unknown, but it is related to the high thermal and chemical stability of the MT-NDPK. In phagocytosis, a phagocyte envelops a bacterial cell by binding to the cell with its receptors. The bacterial cell then becomes engulfed in a vacuole called a phagosome. A lysosome then fuses to the phagosome, becoming a phagolysosome and providing it with the hydrolytic enzymes needed to digest the cell. The remaining molecules are then ejected from the cell.
So why does MT-NDPKs exhibit these high levels of stability? One answer is the salt bridge. Fundamentally, protein salt bridges are electrostatic non-covalent bonds between two oppositely charged amino acids which form at least one hydrogen bond with each other. They are most commonly constructed from the negative charges from aspartic and glutamic acid and the positive charges from arginine and lysine. However, other amino acids with similar properties such as histidine, tyrosine, and serine can also be used in the right conditions. You can see here a depicted salt bond between a lysine and glutamic acid. There is an electrostatic attraction between the positive end of the lysine and the negative charge of the glutamic acid. An important factor for the formation of salt bridges is the proximity between amino acids, as the distance must be less than 400 picometers in order for salt bridges to form. If they are farther apart, the attraction force lessens so that the formation of the bond is not possible. Salt bridges are dependent on the pH, in which a higher pH would lead to deprotonation of the NH3 ion, while a lower pH would lead to the protonation of carboxylate to a carboxyl group. Because of the strength of the salt bridges, the researchers hypothesized that if they mutated R80, the stability of NDPK would decrease. However, the researchers unexpectedly found that the enzyme was still stable due to compensatory factors which allowed the kinase to maintain function. In the next presentation, we will go over more in detail into what the researchers found, the compensatory factors, and the function of the kinase following the R80A and the R80N mutation.